Greetings of the day to everyone. Today, I, Dr. Ansh Chaudhary, is going to discuss something very important with you all, and that is neurological perspective of gut microbiota. First of all, it is very important to understand that the human body is a super complex ecosystem containing trillions of bacteria and other microbes. And these microorganisms are an essential ecological community of symbiotic, commensals, and pathogenic microorganisms which share our body spaces. The role of microbiota is rapidly emerging in gut-brain communication, and alteration in composition of these microbiota have been observed as an important mechanism of several diseases. The coexistence of stress-related nervous system disorders like anxiety, depression, autism, schizophrenia, bipolar disorders, and other degenerative disorders like Parkinson's and multiple sclerosis, etc., is well established, especially with gut disorders like irritable bowel syndrome and inflammatory bowel diseases. Let us now throw some light on mechanisms of gut microbiota, in which we all know that stress is the biggest trigger, along with injudicious use of antimicrobials. All of them influences the microbiota and causes the disease state. The first being dysbiosis. The stress-induced hypomotility of stomach and small intestine results in bacterial colonization and also simultaneously decreases level of beneficial lactobacilli and bifidobacterium. Second one is immune activation. Stress stimulates colonic stimulation and increases innate immunity by production of cytokines by the immune cells. They act as regions of CNS involved in the HPA axis and which further modifies the neuroendocrine activity and the behavioral response. The third one is alteration in the HPA axis, that is the hypothalamic pituitary adrenal axis. Gut-brain axis is composed of four major signaling pathways, that is neuronal, enteroendocrinal, neurosecretory chemicals, and finally the immune system. Persistent stimulation of the HPA axis lead to two things. The first one being release of glucocorticoids by hypothalamus and release of adrenaline and noradrenaline via sympathetic branch of the ANS. All of these lead to dysregulation of immune system and cell-mediated immunity. Let us now talk about gut microbiota and the neuropsychiatric diseases associated with it. Normal gut microbiota plays a vital role in development of brain, especially with myelination of the processes of prefrontal cortex. Absence of colonization of these microbes result in altered gene expression and turnover of various neurotransmitters both in enteral and central nervous system. First of all, let's talk about role in anxiety. Serotonin, a neurotransmitter produced by both brain and the enterochromaffin cells of the gut, is closely related to alter the mood. The host microbial interaction leads to an increased plasma serotonin level to as high as 2.8 folds. Campylobacter jejuni, a common gut microbe, increases anxiety-like behavior by inducing C-phos protein expression in amygdala, hypothalamus, and the stria terminalis. This designs the basis of concept that the antidepressant drugs work better with dietary modification. Second thing is role in depression. Depression is associated with stress, GI disorders like ulcerative colitis and Crohn's disease, which is speculated to be caused due to an imbalance between sympathetic and parasympathetic discharge from the CNS. The altered gut population of Acinetobacter and Bacteroides is observed in individuals with depression. It has also been suggested that the toxins liberated by these bacteria decrease the level of neurotransmitters like 5-HT and brain-derived neurotropic factors that may be responsible for depression. Gut microbiota and the autistic spectrum disorder. Autism is a neurodevelopmental disorder of early childhood associated with deficit in social communication and imaginative development simultaneously. With restrictive activities, repetitive behavior, and intellectual disabilities, GI problems occur in parallel with the behavioral symptoms. More in autistic children. Significant colonization with distinct clostridial species, especially Clostridium sporogens, Clostridium tetani, aggravates the autistic behavior. Treatment with vancomycin causes substantial but transient improvement in autistic behavior, probably due to the effect of antibiotic eliminating the neurotoxin produced by bacteria. This indicates colonization or action of neurotoxic produced by Clostridial species to play a role in autism. Now let's talk about gut microbiota and Parkinson's. The severity of motor movements is related to gastropoiesis, which occurs frequently in Parkinson's disease. Increased abundance of enterobacteria were found to be prevalent in postural instability with gait, phenotype of Parkinson's disorders. Then the tremor dominant type. In H. pylori is correlated with developmental motor functions and Parkinson's disease. It causes intense neurotrophic response and resultant increased muscle contractility. Now, let us talk about gut microbiota and multiple sclerosis. In relapsing remitting multiple sclerosis (RRMS), several organisms like bacteroids, which are primarily involved in bile acid metabolism and metabolism of phytochromogens, were found to be lesser in number as compared to normal population. The metabolites of bile acids and phytochromogens produced by intense anti-inflammatory response, which may be responsible for autoimmune etiology in relapsing remitting multiple sclerosis. Now let us talk about how to target these microbiota. The main method is modulation of diet. The most effective way to modulate the composition of gut microbiota is through a balanced diet. This balanced diet comprises of meat, fish, fruits, cereals, legumes, vegetables, and olive oil, commonly known as the Mediterranean diet. This diet is most efficient to promote optimal gut microbiota composition. This diet benefits by resisting intestinal permeability, reducing endotoxin, and is related in consequences to gut-brain axis with an overall positive microbial effect. Therefore, a healthy lifestyle based on diet as foundation is probably the best way to prevent and treat different disorders associated with disturbances in gut microbiota. I would like to conclude my presentation by saying that gut microbiota has significant impact on brain development along with myelination process. The present presentation points towards the institution of probiotics or aloe vera as an alternative strategy along with judicious use of antibiotics, which will help maintain the perfect balance in the gut microflora. Thank you.